Hello and welcome to our video about lichen.
Over the past few months, we've been conducting surveys across the Dartmoor National Nature Reserve to determine the distribution of 3 species of lichen, while also analysing atmospheric and air quality data to look for trends in Dartmoor over the past few decades.
So, why is this important?
Our lichen species of interest are all sensitive species which are thought to be in decline, and their presence can act as an indicator of the air quality in a habitat. By surveying their distribution and abundance across Dartmoor and analysing the atmospheric and air quality data available in the region, we hope to contribute to the ongoing research in this area.
So, what is a lichen?
Lichens are symbiotic organisms comprised of a photobiont, which is an alga or cyanobacteria, and a mycobiont, which is a fungus. The phycobiont produces carbohydrates via photosynthesis. Some of these carbohydrates can be utilised by the fungus.
In return, the fungi forms a protective cortex around the phycobiont. This protects the phycobiont from various environmental stresses, such as UV light. The fungus also provides water and minerals to the phycobiont.
This strong mutualistic relationship allows lichens to colonise areas such as rocks, wooden gates and tree branches that they may be unable to do as a single organism. As such, they are often pioneer species in rocky environments. This means that they are important in succession, the evolution of an ecological community over time.
Growth and colonisation of areas by lichens is dependent on various air quality factors such as nitrogen oxide, ozone and sulfur dioxide. Specifically, Bryoria fluorescens has been shown to be negatively affected by acid rain, heavy metals, ammonium and sulfur dioxide. Therefore, we would expect that the presence of these factors should cause a reduction in occurrence of our lichens of interest.
Our project starts with a survey of various tors and woodlands in Dartmoor for the presence of Bryoria species, fluorescens and bicolor, and Usnea species, articulata and florida, in order for us to determine the current status of these lichens in the region.
The data collected was used to satisfy our 3 main objectives. Firstly, to get the first quantitative description of Bryoria populations across Dartmoor and compare current populations to those described in a paper published nearly 30 years ago. Secondly, to compare all Usnea transects carried out previously and compare these to our own. Any trends in the data would be analysed. Lastly, to visualise and analyse air quality and atmospheric data available for Dartmoor.
In our surveys of Bryoria, we visited a total of 10 sites across Dartmoor, including tors, old stone walls and woodland. On each trip, a group consisting of a species identifier, a note-taker and a photographer systematically searched the site for the presence of Bryoria, marking the GPS location of every spot on which Bryoria was found. The location, size and positioning on the substrate were recorded for each individual found. Accessibility was a barrier to comprehensive surveying of some sites due to vertical rock faces, sizable crags or dangerous ground underfoot.
Getting more and more confident in these rocks. I can tell you.
Yeah.
So to give a clear picture of everywhere we surveyed, we also recorded null results where we found no Bryoria.
Nothing. We have 4 different group sizes for classifying the Bryoria. So here you can see a very small one, so that would be group 1, which is 0 to 1 centimetre. Here you can see another group which is slightly bigger, so that would be group 2, which is 1 to 2 centimetres. This one's a bit bigger, so this would be a group 3, 2 to 5 centimetres. And this humongous colony here would be a group 4, which is 5 centimetres plus.
In the second stage of our investigation of Dartmoor's lichen, we carried out surveys along a selection of Usnea transects with the aim of locating 2 species of Usnea, Usnea florida and Usnea articulata. Transects are specific routes that are followed to monitor the presence of a particular species. We walked these routes marking the GPS location of Usnea on fallen twigs or in overhead branches on trees.
While our most active data collection was coming in from the trips out to Dartmoor, back in Exeter work was ongoing to harvest the records kept by DEFRA from the Yarnwood Air Monitoring Station. This dataset covers over 30 years of measurements, providing a comprehensive record for pollutants such as nitrogen oxide, nitrogen dioxide and ozone. Putting this data to good use was quite a challenge. It produced a spreadsheet with over 250,000 rows and required us to learn a new programming language, R, to be able to analyse it effectively. Thankfully, King's College London had a fantastic software package and guidebook for exactly this purpose. Using the open-air library in R, we were easily able to analyse and produce graphs to visualise our findings.
Our most surprising discovery was that over the 30 years that the data cover, there is very little long-term change for the atmospheric concentrations of these pollutants.
Ozone data was available from 1987 and shows levels have remained pretty much constant at around 60 micrograms per metre cubed. Conversely, nitrogen oxide and dioxide both showed a 4% decrease per year since measurements began in 2003. This is despite a huge increase in the volume of traffic on our roads and may be due to the relatively remote location of Yarna Wood, helping to isolate it from the worst of vehicle emissions.
There is still some influence on these emissions at shorter timescales. It was interesting to see quite how sensitive the system was by plotting an average weekly cycle. For the nitrogen-based pollutants, it's very clear how closely linked these are to car exhaust emissions, with peaks corresponding to peak commuter times past the monitoring station. These show that on the weekdays, levels are elevated for nitrogen oxide and dioxide, with a noticeable double peak for nitrogen dioxide that is absent for nitrogen oxide. These peaks occur daily, but values return to a baseline overnight. Ozone levels are essentially constant regardless of the day of the week, peaking during the day when UV radiation stimulates ozone precursors to react.
So, while this was interesting data, it did not help us much in explaining the distribution of lichens that we had discovered. In part, we believe this is due to the limited scope of the monitoring site, only giving us data for a single point on Dartmoor. Because of this, we deduced we would have to look into the specific altitude and aspect of each site we visited for sampling, particularly with regards to bryoria. Thus, it appears local factors are the predominant influence on distribution of lichens, rather than larger trends in air quality.
In total, 10 sites were surveyed primarily for the presence of bryoria. However, only 9 of these are shown as quantitative data could not be obtained for one of these sites. The coloured circles show exact GPS locations. An adapted ACFOR scale based on the total length of bryoria, measured by tough number and length, was used to colour code their relative abundance. This is shown in the top left of the figure. One of our objectives for the bryoria surveys was to compare the current state of bryoria populations to those observed in 1990, described in this paper. However, only 3 of the sites we visited were mentioned in this paper: Bear Down Tor, Blackingstone Rock and Sharp Tor. At Bear Down Tor, bryoria bicolour was found in 1990, but are now absent from this site, and weren't found at any other of the sites. In fact, at Bear Down neither bryoria species were found at all.
At Blackingstone Tor, the paper noted the presence of bryoria faucescens in narrow cracks in the rock face, which were stunted in size. In our survey, we found only 3 small tufts, which seems consistent with Giavarini's findings, and hints that although this tor is not a favourable habitat for bryoria, there has not been a significant change in the population here.
At Sharp Tor, 53 bryoria tufts were found across 3 locations on the site, all on the eastern side of the tor, in close proximity to one another. Many of these were very large clumps, whereas in the 1990 paper only a few tufts were found, and these were described to be in poor condition. It seems that bryoria is flourishing at this site.
So, for the sites which we have historical data to compare to, we have not observed any consistent trends in the change of bryoria populations across Dartmoor. Instead, we decided to look into the possible effects of altitude and aspect on the prevalence of bryoria, since there is such a variation in the quantity and size of bryoria found at each site.
One local factor that we thought may influence abundance of bryoria was the altitude of the site. Abundance was measured for each site by multiplying the number of bryoria assigned to each size category by their approximate length. This produced values representative of the total length of all bryoria tufts placed end to end for each site. There does not appear to be any correlation between altitude and the presence of bryoria. It is still possible that there is a link, but since data was analysed for only 9 sites, a larger survey across Dartmoor would be needed to give a comprehensive insight into this relationship.
To analyse aspect preference, the length of all bryoria tufts facing each direction was totalled for all of the sites. Our data suggests a strong growth preference to the northeast and the southeast, as well as a weaker preference for growing to the east. This was thought to be due to light intensity. The average light intensity per day on a bryoria tuft facing eastward will be much less than a tuft facing southwards. This means that they are exposed to less solar radiation on the east-facing side of the boulder, enhancing growth and survival. However, various other factors such as wind direction may also contribute to this trend. Therefore, to determine whether light intensity is a major factor causing this trend, controlled laboratory experiments could be conducted.
Next, we looked at Usnea, the other genus of lichen. This involved combining data we collected with existing data from previous years. A number of challenges became apparent as we searched for trends in data. Previous surveys were inconsistent, some specifically looking at one Usnea species and others not giving exact numbers of individuals found at each point along the transect. Another problem was that there was a large covering of leaves on the canopy due to the mild weather we've had this autumn. This may have prevented us from identifying some of the Usnea present.
We plotted all the transects we surveyed onto one map, with markers representing the Usnea individuals found. From this, it is clear to see that Usnea are flourishing on certain transects and not others. You can see that high numbers of Usnea were found on transects 2, 4, 5 and 9, whilst none, or only a few, were found on transects 3, 6 and 8. Zooming into transects, we can identify specific hotspots. For example, 43% of Usnea florida along transect 5 was found in the northern part of the transect near Needham-Cleeve. A graph was made for each species, showing the number of individuals found each year along each transect. It's important to note that where you see NA, we didn't have any data for this year. You can see that there is a huge difference between transects in which Usnea species grow, possibly due to one species outcompeting the other. For example, on transect 2, Usnea articulata are thriving, with 330 individuals found this year. However, only one individual of Usnea florida was found. In contrast, more Usnea florida than articulata were found on transects 4, 5, 9 and 10. Furthermore, numbers of articulata were much higher on transect 2 than on all the other transects.
Now looking at population changes over time, you can see that on transects 2, 3, 4, 5, 6 and 9, there has been a decrease in the population of Usnea florida since 2011. Along transect 8, it appears that Usnea florida have struggled to establish themselves and have been rarely identified. On transect 10, we did not have any data for the years prior to 2016 and 17, so it was difficult to draw any conclusions about population changes here. However, numbers are quite low compared to the other transects, suggesting it isn't an area where Usnea species can thrive. It is worth noting that on transect 4, although there is a decreasing trend, the significant drop in numbers from 2012 to 2014 suggests that the whole length of the transect may not have been surveyed in 2014. This can also be seen on transect 6, where a dramatic drop in numbers counted between 2012 and 2014 may indicate an issue with the surveying quality. Furthermore, along transect 5 in particular, the numbers of Usnea florida may be underestimated, as the height of the trees made it hard to spot certain species and identify them with certainty, so the majority of the Usnea florida found were on fallen material.
Next, we looked at temporal changes in Usnea articulata. It appears that the population of this species has been thriving along transect 2, with a gradual increase in number of individuals from 2014 to 2017. Interestingly, for transect 3 and 6, Usnea articulata has never been identified, only Usnea florida, which suggests that it's not favourable for this species to grow in this area. You can also see that Usnea articulata has also been found in very low quantities at transects 4, 8 and 10. The presence of Xanthoria at the end of twigs along transect 8 suggests a nitrogen-rich environment at this site, perhaps explaining the lack of Usnea species. Looking at transect 5, numbers seem to be increasing here, however, no individuals were identified during our survey. We'd have expected numbers to be lower due to the amount of leaf coverage, however, it is unusual that we didn't identify any individuals at all. And finally, looking at transect 9, there doesn't appear to be any overall trend in abundance over the years.
Overall, we found that there were no significant changes in atmospheric data that may be correlated to lichen abundance in Dartmoor. When comparing our Bryoria surveys to Giovanni's discussion on Dartmoor's lichens, we found no evidence of a species decline. In an attempt to instead correlate Bryoria distribution across sites to altitude and aspect, we found no correlation with altitude, but a possible orientational preference to southeast and northeast. Trends in Usnea populations weren't very consistent, although Usnea florida does appear to be on the decline.
Future atmospheric analysis could involve utilising other data available from the Dartmoor monitoring station, such as sulphur dioxide. This work could also be built on via repeated sampling of a larger sample of tors across Dartmoor, utilising the quantitative surveying method we devised. Further investigations could be done into the effects of local factors such as controlled experiment on the orientational preference of Bryoria. And finally, we would recommend gathering consistent Usnea data each year at the same time to identify trends more clearly.
We found no lichens, but it's okay. It's Jenny. It's a fun rock. Wait, woo! Glad there's ferns everywhere. There's just nothing there. What are you standing on? How do you get down here? How did you do it? I'm coming. The film you He's coming.
I don't know whether I'm coming or going at the moment.
It's like a slide.